muscle, generally we have muscles that are attached to bone by tough tendon sheaths, and so these are due to connective tissue. So when a muscle contracts, it's going to shorten in terms of skeletal muscle. This creates tension on tendons that's connected to the bone, and this moves the bone at a joint. So that's why you'll have the crossover of the tendon that goes over that joint so that we're able to move that joint. And the bone that moves, that is attached, is at the muscle's insertion, and the bone that does not move is attached to the muscle's origin. So movement tends to be towards the insertion point of a muscle. So the movement of our muscles and bones depends on the joint and how the muscles are attached. So there's two main categories, the flexors and extensors. So flexors are going to decrease an angle between two bones at a joint, and extensors are going to increase the angle between two bones at a joint. So the main muscle that is responsible for movement in a direction is going to be an agonist, or is going to be a prime mover. And muscles that work in opposite actions are going to be antagonists. So flexors are antagonistic to extensors and vice versa. So there are three main layers when we look at skeletal muscle in terms of its connective tissue. So the first one is the epimysium, and the epimysium is going to be the outer layer that surrounds the entire skeletal muscle. Then we have the perimysium, and that is going to surround individual fascicles. And so the last layer that we have is the endomysium, and that's that will surround individual muscle fibers, or the myofibrils of connective tissue. Muscle fibers, just like any other cell in the body, has the same organelles that we find. So like they have nuclei, they have mitochondria, and for instance, they have something though a little bit different than the endoplasmic reticulum. Rather, they have a sarcoplasmic reticulum. And so they also have a special plasma membrane, however, same function, but it's just called a sarcolemma. Muscle fibers also tend to have lots of nuclei, and so here in this image, what we're seeing are those dark stains, and this helps to form a syncytium, where this means that they contract as a unit. Another important feature about muscle fibers is that they have different bands. So they have this I band and the A band, and so I is standing for the light bands of only actin, and then A bands stand for the dark bands of myosin. And so when we look here then at the muscle fiber, we see different striations. So the darker purple representing the A band, and then the lighter representing the A band. And what we also have too, where is the definition of one unit of the muscle fiber, is dependent on the Z line. And so that's going to make the sarcomere. And so this is where the dark lines in the middle occur within the I band. And so one contractile unit is called a sarcomere, and that's based from Z disc or Z line to Z line. And those are going to span the entire length of the skeletal muscle fiber. So when we look at the neuromuscular junction, here we're looking at histology or staining of an actual neuromuscular junction within the rat. And so axons of these motor neurons are going to be myelinated because they're going to be larger, and also they tend to be farther away. Think about like moving your big toe. That is a long distance for that axon to travel, and so in order for that impulse to go quickly, they are myelinated. Also, neuromuscular junctions tend to have many terminal branches, and so this is what we're seeing here, where it's branching off from that main axon terminal. The synapse between the neuron and the muscle then is going to be the actual neuromuscular junction. And the main neurotransmitter that will be released at the neuromuscular junction is going to be acetylcholine. And a special note too is that skeletal muscles only have nicotinic acetylcholine receptors, and so these are always going to be excitable. Okay, so whenever we have release from the neuron of acetylcholine, and acetylcholine binds to nicotinic acetylcholine receptors, that's going to activate that skeletal muscle and make it excitable. So here we have the events that are occurring within that neuromuscular junction. Okay, so let's start at here. We have the action potential is being conducted down that axon, and it's going to invade into the axon terminal. Okay, so what will happen then is that that change in voltage is going to activate voltage-gated calcium channels. And so if you remember, calcium is in higher concentrations outside the cell than inside. So based on diffusion properties, it wants to enter into the cell. Okay, so calcium is going to rush in, and within that axon terminal, what we find here are these little vesicles of neurotransmitter. And again, at the neuromuscular junction, these are going to be filled with acetylcholine. Okay, so the synaptic vesicles, these are either going to be towards the top here, or there's going to be some that are going to be docked and stationed and ready to go at the end here, okay, by the plasma membrane. So then by exocytosis, what will happen is that the synaptic vesicle will fuse with the membrane, and then it's going to release acetylcholine out into the synaptic cleft. Okay, so then once acetylcholine binds at the motor end plate, now we're looking at skeletal muscle here, this will actually bind to these nicotinic acetylcholine receptors. Okay, and what will happen then is that sodium is going to rush into the cell because sodium is higher outside than inside, and then potassium is going to leave the cell. So this is going to cause depolarization, all right? And so what happens then with the motor end plate here, which is lined with lots and lots of these different nicotinic acetylcholine receptors, is that that change in depolarization then is going to cause an action potential that occurs within the skeletal muscle that's going to cause it to contract. Well, about as soon as this happens, then we also have swift removal of all this extra acetylcholine that hasn't bound by acetylcholine esterase here. And so that's going to break it down so that our muscles don't stay constantly contracted and excited. Otherwise, that would get into tetany, which would be very painful over a long time in a living individual. So neurons and muscles are excitable cells. And so when we have the two together, what we have are motor units. And so a motor unit is a single motor neuron and all of the muscle fiber innervates. And thus, all the muscle fibers in a motor unit then are going to contract at once, providing that syncytium. This will create graded contractions, which are going to be varied contraction strength. They'll be due to different numbers of motor units being stimulated. So in larger muscles, like say biceps or the hamstrings, they'll have a larger motor unit because they have a lot more muscle fibers that are being recruited compared to something as fine as like your index finger, where that will have a smaller motor unit because you're activating a less number of muscle fibers. So the neuromuscular junction, which is here, is going to be the site where the motor neuron is actually going to touch with the muscle fiber, and that's where it will also release its neurotransmitter of acetylcholine, making that skeletal muscle excitable. And so the region of the muscle fiber where the motor neuron is going to stimulate is going to be called the motor end plate on the muscle. So with control of motor units, there's going to be either one of two things. Either it's going to be for fine control or for strength control. And so with fine control, this is when we have smaller motor units that are going to contain as few as 20 muscle fibers per nerve fiber. And so we see this with the eye muscles, and so this allows for greater dexterity because of a lower neuron to muscle fiber ratio, okay? However, the strength control is when we have one motor neuron controlling many muscle fibers, and that allows for greater strength production because of a greater recruitment. And so, for instance, this is with the gastrocnemius, and that can have about 1,000 fibers per nerve fiber. So there is a fine control versus a strength trade-off for the two.